Thank you, gentlemen.
Hey, thank you very much, Linda, for that introduction. And Christiana, really appreciate you, your assistance too. So it's a pleasure to be here this evening. And I'm just adjusting my camera so you can see me better. And so Dan and I have had a lot of experience in the past couple of years, actually, with cell and gene therapy, which are also called in Europe ATMP facilities. And we basically have drawn upon that experience in the industry to produce this talk. And we've been giving different variations of it to ISPE, and I thought it would be great to give it here this morning for you and to answer any questions. So with that said, we're going to take a deep dive into this topic, and the focus of it will be a risk assessment approach to address fungal spore contamination in cell and gene therapy clean rooms. So let me go ahead and move to the first slide. This is just my background and experience. It's 21 years worth of industry experience. And nothing beats experience, and my colleague Dan Klein also has 20 years of experience. So I think with that, that's kind of almost 40 years of total experience here on the board with you. So we should be well suited to provide some good background on this topic tonight. So to begin with, I think it's helpful if what we do is talk a little bit about the most common contamination sources in the industry. Regarding the new Annex 1, we are now on version 13, and they're expected to actually release the final version of Annex 1 in the first quarter of 2022. So that's the plan in the industry, and then the plan in the US will be, from what I'm hearing, they're going to be taking up the aseptic processing guide, which was put out by the FDA in 2005. And just like Annex 1, the industry is going to basically start to rewrite that and update it very similar to our Annex 1. So speaking about contamination control sources, some of the most common sources in the industry are the facility, so aging facilities, the type of facility, the type of production you do in the facility. All of those can lead to potential bioburden contamination. Materials, this is a big one. Dan and I really like to talk about this. Any items you're passing through into the clean room, so intervention tools, bags, items in bags, equipment, hammers, anything like that, carts especially, and cart wheels that are coming from one clean room class to another are a huge source of mold spores and bacterial spores and a potential risk of contamination in the clean room facility, and especially in cell and gene therapy. When it comes to personnel, operators, that's your #1 source of bioburden contamination in any clean room. We shed millions of particles and millions of skin flakes a day. And even if you gown up to go in a clean room, you are still going to have particles and skin flakes that will flake off and get on the surfaces in the room that can harbor viable microorganisms. And of course, whatever process you're doing in that clean room, if you're doing tableting, if you're doing cell and gene therapy, if you're doing work with roller bottles and media, or blood fractionation and cold temperatures, all of those are a potential risk for spreading contamination and spreading residues and particles in the room. And then, of course, utilities. So water systems like WFI systems, compressed gases, helium, nitrogen, argon, those are all potential risk for contamination. One of the biggest organisms we hear a lot about in the industry from compressed gases are anaerobic bacteria such as Cutibacterium acnes, which can live in an anaerobic or aerobic environment, and it comes from operators with bad acne. So they'll touch their skin and they touch the surface in the room and they spread that bioburden. And then, of course, equipment and where you place that equipment in a clean room is always a potential risk when it comes to contamination. So before I go to the next slide, why don't we go ahead, Linda, and bring up one of the polls, the first one. You want me to read it, Linda?
Yeah, it should be coming up on the screen.
Okay, I see it here. So the poll is, does your facility have a CCS, which is contamination control strategy? That's something mentioned in Annex 1, in place. So the choices are A, we are developing one. B, we have one in place. C, we need more information. Or D, we don't have it at this time. And we don't know what it is. Which of those do you choose? And there's actually more than one right answer here.
So we're just waiting for everyone to put in their answer.
Okay.
And then we'll show the results shortly.
Sure. I'll hold off before going on until we get the results on that. All right. So we need more information is the number one thing. And that could be possible. So my advice on that would be, I would make sure to get the latest copy you can of Annex 1 version 12, which is out there, or to get the latest annex before that that was published. I believe it was in 2017. And then, as I mentioned, we are on version 13, and the finalized version should be coming out. But contamination control strategy covers many different facets of contamination. So not just cleaning and disinfection, it covers endotoxin, it covers the facility design, operations of the facility. So about 10 different sections are included in it. So I would highly encourage you to take a closer look at the annex and take a look at that, because that will be something, if you're selling products in Europe or you have a manufacturing site in Europe, that's something European inspectors will expect you to either have or at least be developing and formulating one. So those are the top three answers here. We need more information, we have one in place, and then we are developing one. And I think those are all pretty good, suitable answers. So let's move on to the next slide here. Let me get this going here. I always find this slide very helpful in the industry to kind of talk through this. And that is when we look at a contamination control strategy and we get very specific with it, and we talk about the not just the CCS, but the aspect of cleaning and disinfection and microbial control, it's really critical to think about what disinfectants and what sporicides you're going to be using. So in terms of that, when selecting a disinfectant or sporicide, it's very critical to think about the key attributes that you want. And if you take a closer look at this, this is a delicate balance. You want to look at things like efficacy. So my good colleague in the industry, Fred Ayers from Lilly, would say, if it doesn't kill my bioburden, I'm not going to be interested in it. And it has to be compatible, because it's in his insulin production area. He doesn't want a product that's going to destroy his stainless steel. Stability, and FDA does, believe it or not, ask about stability. So use dilution stability, open container stability. Those are two things that end users in our industry get asked very frequently and commonly. Safety. So is the product safe for my operators to use? Is it safe to spray? Safe to apply with mops and wipes? And then a big one can be disposal. Is it something that I can send straight to drain to any sanitary sewer system? Or are there restrictions such as on pH or phenol content? And those are questions that your local water authority should have feedback for you on. Rinsing. So is it something that can easily be rinsed with water for injection or 70% IPA? Can it easily be removed from the surface? And is it something you can remove periodically quite easily? So let's talk about some of the cell and gene therapy case studies. There have been several. So even at the time we put this together, there have been several that we've had in the industry even since then. So let's take a closer look here at the cell and gene therapy process. And if we do that, the typical process, and there's many steps in this where I'm going to talk about using a disinfectant or a sporicide. The first process is you bring in that core blood from a patient or patients, and it will come into the clean room in a cooler. So from outside ancillary areas, it comes into the ISO 8 room or grade D area. And in that cooler, you can wipe it down with a sporicidal wipe, or you can spray it down with the sporicide and then wipe it. But that cooler is a key thing because that's going to have that core blood in it that I'm going to be bringing then into the clean room to use in making my cell and gene therapy products. And this is a real exciting area in the industry today, because some of the cell and gene therapy sites I've been working with are working on cures for some key diseases out there, such as diabetes, Huntington's disease, congestive heart failure, blindness. And I can see in the next, say, 20 years, some of these products are going to come to fruition and come out in a marketplace, because insurance companies, instead of spending millions of dollars a year for insulin for patients, if I can give one shot to a patient and I can cure the diabetes, it's a godsend. So how does the process work? Well, I take that core blood, I bring it into that ISO 8 area, ISO 8, which is typically my gowning room. And then from the gowning room, I bring it into the clean room, the ISO 7, which is the area right outside of my BSC hood. In that area, what I'm going to be doing is isolating the T cells and the natural killer cells. And the way that works is I'm going to be doing that process in a BSC hood with high airflow. So in these hoods, I'm going to be adding a receptor and a protein to it. And then I'm going to be taking those cell lines, and I used to do this kind of work in a biotech lab, and I'm going to be growing up those cell lines in an incubator. So two of the key things to think about with the incubator are there's a lot of problems with mold in the incubator. Mold can start to grow in those gaskets. So you want to make sure to take a sporicide or sporicidal wipe and wipe down the gasket area and the inside of the incubator and outside. A sporicide is a good product to use there. A lot of it is made of stainless steel because you want to make sure that you don't potentially have any viruses that could infect your cell lines or any bacteria. So the incubator is a big source here when growing up the cells. You want to make sure there's no potential bioburden contamination in there. Then I'm going to be harvesting and washing those cell lines, right? I'm going to then be filling bags and vials with them. And then I'm going to be taking those bags and vials outside of the ISO 7 clean room or grade C area into the grade D. And then even outside of there, maybe in some of the CNC, controlled not classified areas, and cryo freezing it to minus 80 or below. And then depending on what type of cell and gene therapy, whether it's allogenic or autologous, I'm going to be loading it into a cryo shipper and shipping it either to a single patient or to multiple patients, depending on what type of cell or gene therapy I'm doing. One of my good colleagues in the industry that does a lot of this work is Irving Ford with BMS, and they are constantly doing this kind of process. Two of the key products he always promotes there, the sporicidal wipes and use of the sporicide in the BSC, and ready-to-use products in there. So ready-to-use disinfectants like quaternary ammoniums are a great product to recommend inside these BSC hoods. You can use phenols too, but I would say the quats are very easy to use, and they do come out there. There are some with ready-to-use formulas that have enhanced efficacy against molds. So then talking and a little bit about the process and where we can come in with these kind of chemistries. First of all, with any of the gloves, so gloved hands doing work and manipulating the cells inside the BSC hood, which is the ISO 5 area, I can use 70% aerosol alcohol on them. I can spray them down with a trigger spray alcohol that's sterile. And keep in mind, anything used in that BSC hood should be sterile or at least filtered before using in there if it's not sterile. The sporicidal wipes are great to use for wiping down the walls and the floor inside the hood. I think that's something I recommend a lot in the industry. It's very popular. And of course, any items coming in there. So if you've got little tweezers or you've got vials you're working with the cell lines inside there, wiping them down with the sporicidal wipe is a very good process to undertake. And then, of course, as I mentioned, the incubator, and especially getting that gasket. The gasket's going to have ribs. You can separate the ribs of the gasket and get in there to the nooks and crannies with the sporicidal wipe to address any mold contamination there. So just showing you some of the data here, and I want to talk through this because this is important. On stainless steel, and this is data we published in a PDA book chapter, we want to look at Aspergillus brasiliensis, a very common mold in cell and gene therapy. So we wanted to look at different surfaces, typically that could be pass-through items into the clean room. One would be stainless steel. You'll see it on carts. You might see it on intervention equipment. And if you have active air samplers, a lot of times the head of them is made of stainless steel. So what we're showing here in terms of contact time, we're showing efficacy with 70% IPA and then a sporicide, which is a hydrogen peroxide peracetic acid chemistry, and then the sporicide plus the IPA, where we actually see a bit of a synergistic effect. So if you look at this, we have a baseline inoculum, and then we're seeing log reduction numbers, very high numbers with sporicide and the sporicide plus the IPA of greater than 4 log reduction. And keep in mind, USP 1072 requires you to have at least a 2 log reduction of bacterial spores. So you're seeing that in a very short time. So keep in mind, this is just 5 minutes, 6 minutes total when I add in the IPA. So we're seeing nearly a total kill. Very good data. We're seeing the same thing with the self-seal pouches. So this comes from our Barrier Product Systems group. And when you look at this with the self-seal pouches, again, sporicide plus isopropanol, again, greater than 4 log reduction. So nearly a complete kill. Very good data to share with the customer. And it's all published data. The same is true with Lexan, which is also the trade name is polycarbonate. So again, a sporicide and sporicide with IPA greater than 4 log reduction. And again, nearly complete kill. Very good data that customers would be interested in. So if I go back a slide here, another thing I just wanted to mention here, for your general control of bioburden in the ISO 7, ISO 8, which is grade C and D clean room, and the CNC area, one of the best products to use is quaternary ammoniums. So there's ready to use versions out there, and there's also phenolic disinfectants. And there's a very close to a ready to use version of that. Then you really do need to have periodic use of a sporicide, because one of the things Dan and I have seen a lot lately are some of these cell and gene therapy sites that have become completely contaminated with penicillium spores. That's a mold spore that is fairly hard to kill, not the hardest, but certainly not the easiest to kill. And so hydrogen peroxide peracetic acid chemistries and bleach at 0.525%, which is 5,250 ppm of available chlorine, would be the kind of chemistries you would want to use to control spores. And then, of course, IPA. That's great for rinsing and residue removal. It's also a great technology if you want to sanitize any items going into the BSC or any items coming into the clean room. So that's a key thing, is your BSC hood, frequent use of a sporicide in there. And then you're going to need a rinsing step with IPA or WFI, and then wiping it to remove any potential particles or residue so that doesn't get into your cell and gene therapy products. So moving up a couple slides here. When we look at this one, we are looking at a very hard to kill bacterial endospore. So it's Bacillus subtilis. This is used in the US EPA's sporicidal challenge test, B subtilis. And again, we're looking at stainless steel. In just 5 minutes, you're seeing almost a 2 log reduction. And then with that, you see the synergistic effect with the IPA, 1.72 log reduction, so 98% kill, very good. With the self-seal pouches, with the sporicide, 1.47, sporicide with IPA, 1.98. So pretty good log reduction, again, in just 5 minutes. So most pharma biotech companies with a lot of the sporicides are looking for a 10-minute contact time. And to be honest with you, in ISO 7, ISO 8, you can easily get that. And some of the BSC hoods, it would be challenging, so you may need to reapply. So down here with the polycarbonate, again, 0.68 log reduction, and you're seeing 1.25 log reduction with the IPA added. So that's synergistic effect. And again, you know, some fairly decent percentage of kill again. So what are some of the best products to use in these cell and gene therapy facilities, which in Europe, again, they call them ATMP, Advanced Therapy Medicinal Products. So when you look at that, wipes, so hydrogen peroxide peracetic acid or hydrogen peroxide wipes, they are great disinfectant wipes. They do kill spores. So that's something to keep in mind, especially for your hood. And we were just at a cell and gene therapy in Memphis, Tennessee. They had about 22 hoods. And in all of those hoods, they're going to need frequent use of a sporicide and IPA for that residual removal. So again, the wipes are something I always recommend there. Some are pictured here. And ready-to-use chemistries. I especially like to see the ready-to-use quats. You can use them in a 1:64 dilution, where you see enhanced efficacy data against hard-to-kill mold spores like Aspergillus brasiliensis, which is ATCC strain 16404, pretty hard to kill strain of fungus. And as you can see pictured here, you can buy sterile or non-sterile. Of course, you would need the sterile in the grade A/B area, which is ISO 5 hood. And here's some additional data that we generated. We wanted to look kind of more so at the clean room itself. So the ISO 7, ISO 8 areas. One of the things we do see when you look at hydrogen peroxide peracetic acid chemistry and bleach at 5,250 ppm of available chlorine, you see very nice efficacy at 1 minute up to 10 minutes against a hard to kill mold, Aspergillus brasiliensis. And one of the things you will notice, 6% hydrogen peroxide takes a lot longer contact time of, let's say, 30 to 45 minutes to build up that efficacy. If it were a higher percentage, it would probably do better, but it's not got near the overall performance. So that's why I normally recommend it as either a very effective sanitizer or a medium level disinfectant. Then we look at Bacillus subtilis, and Bacillus subtilis is a very common bacterial endospore in a clean room. As you can see here, again, with that sporicidal data, hydrogen peroxide peroxyacetic acid, very effective in 5 to 20 minutes. The same is true with bleach, especially at 15 and 20 minutes. And then again, that 6% takes a while to build up its efficacy profile. And here's that data that in more graphical detail with color. And that comes from a PDA poster that I published in the industry a few years ago. A lot of this data was generated in Dan's R&D lab when he managed the R&D group. So let's go ahead and ask that next poll question now, Linda, and then I'll get into the crux of the case study. So with this poll question, and again, it's multiple choice with more than one right answer, is what products does your facility rotate? So the answers are 2 disinfectants and a sporicide, 1 disinfectant and a sporicide, or 2 sporicides, or 3 disinfectants. So let me know what your answer is, and more than one of these can be right. So I give my answer. Let me know when you have something, Linda. I always love a good poll question.
Just give everyone a chance to put in their answer.
Sure.
And then we'll share.
And is Dan still here? He somehow disappeared from the pictures.
You get the spotlight, Jim.
I do. Okay. Well, I'm the show. So. All right. Great answer. So here, 53% of the people that answered the question on the call say 2 disinfectants and a sporicide. Very common in the industry, I'd say. And even more common, I think today, we see 42 of the respondents saying 1 disinfectant and a sporicide. 3% say 2 sporicides. That's very uncommon. And 6% say 3 disinfectants. So the majority of those are what we do see a lot in our industry today, which is 2 disinfectants and a sporicide, or 1 disinfectant and 1 sporicide. So that's what I see a lot of. So great answers. So let's talk a little bit about this case study. So this was a cell and gene therapy site that Dan and I worked with way back at the start of the COVID pandemic. And they contacted us along with Concordia Valsource. So we work with Mark Glavgowski there, industry consultant. They had a huge Aspergillus brasiliensis outbreak going on in multiple clean rooms. This was in the Northeast at a cell and gene therapy site. They had very high environmental monitoring counts in all areas of the site, especially around the BSC hoods. So some questions were asked, and here are some of the answers we received, because one of the things you always want to do is, as Thomas Arista from FDA would tell you, you need to take a deeper dive in to find the potential root causes of the problem. So we did do some of that. So the investigation revealed they were using very excessive water in the facility, and it was potable water. Does that sound like a good idea to use potable water throughout the site, especially for rinsing? Probably not, right? Because of bioburden and potential issues, not just from the bioburden, but even endotoxin and particles. They had the incorrect cleaning and disinfection procedures for the BSC hood. They were just using 70% IPA. Is that the best product to use in these hoods? Probably not, right? Because we have spores occasionally. The tacky mats in the facility appeared very black and dirty, like they had never been cleaned. And if you work with some of these companies that sell the tacky mats, right, like Dyson, they're going to have very specific procedures for cleaning them, more than likely with the disinfectant, with surfactants, and then a rinsing step, but it does need to periodically be cleaned. The mop heads, which were supposed to be white, were all discolored. They were very black in color. So I asked questions about how are they laundered and cleaned? Are they sterilized through radiation or steam sterilization? Then, and this is a big one, we asked questions about the sporicide and disinfectant and how they are diluted. Both the disinfectant and the sporicide were not being diluted correctly. So as an example, the disinfectant was supposed to be a 1 ounce per gallon, so 1 to 128 dilution, and they were diluting it a 1 to 256, so a ½ ounce per gallon. That's a big issue, right? And the same is true with the sporicide. So the sporicide, we would recommend to kill spores to use it at 1 to 32. They were using it 1 to 100 dilution. So those are all potential red flags and causes for concern and things that you're going to want to risk assess. So what are the next steps in this investigation? Well, let's take a look. Well, I always, just like Dr. Scott Sutton would always say, and Tony Kendall, we need to take a deeper dive, and one of the best ways to do that, as my friend Benoit Ramond always says at Sanofi, let's draw that root cause diagram, Ashikawa diagram. It's also called a fishbone diagram. So our source of contamination is the Aspergillus, right? The mold spores. We look at man, so operators, how are they gowning up? Have they been plated? Have they been trained in gowning? How often are they trained? Could that be a source? I was just working with my colleague, Beth Croger, and we have a situation down in Louisiana where we have operators coming in from the swamp area. And now they have an outbreak of Neo's favorite organism, Penne bacillus. So that's a cause for concern, right? How's it getting in there? That swampy area, just like farmland, you can spread that right on into the facility. And that's not an easy one to kill either. Measurement, so how are we taking the environmental monitoring data? Have we got clean data, or is it potentially contaminated? When we pass materials into the facility, how do we decom them? I worked with a facility in Paris a couple of years ago. They had a big bacillus outbreak going on, and I noticed that they had carts outside the ISO 5 area, which is grade A area, and everything going into the clean room from the boxes on the carts was not sprayed down. It went all the way into the RABs. That's a big red flag and a big risk, because you could be spreading any kind of contamination from that. Another big thing to think about there, cartwheels. I remember doing an audit at a facility, Cleanroom site in North Carolina. And they said to me, well, Jim, we have dedicated carts for this area. And then I look out the window and guess what's coming across the parking lot? The dedicated carts. So they're not too dedicated if they're walking across the parking lot. So that's a concern. You should be using a sporicide on those cartwheels, and you should be spraying them down or wiping them down 360 degrees to alleviate any spore concerns, even cleaning underneath the cart, right? So I would take the cart, take it upside down, and I would clean underneath it, potential source of spores, and even the handle of the cart, it's open underneath. So underneath, that's a potential source as well of spores. What method am I using in the clean room for gathering environmental monitoring data, right? What type of production am I doing in there? So, and for example, method, active air sampling, Rodak plating, subtle plating, rapid micro, what methods am I using? When it comes to machine, what type of cleaning are we doing? I work with some sites, like a site out of France, where they're using scrubbers. They ride the little scrubber like a Zamboni, and they ride that in the clean room. Could that be a potential source of Aspergillus? It sure could be. What if you have a mulch pile outside, right? I work with a site where they had a mulch pile outside of the facility in Canada, and then the mold that they found in the mulch pile, which was a strain of Aspergillus, was showing up in the gowning room, in the aseptic area, and then also very critically in the BSC hood where they were doing all their sterility testing. Big concern. Another key tool that you can use in your arsenal when doing a risk assessment and assessing potential sources of contamination in the cell and gene therapy site is the 5 whys. And I can always come up with 6, but let's just talk about the 5, right? So come up with a problem statement. Our facility is contaminated with Aspergillus. Not good, right? So 1 why could be the wrong dilutions of disinfectant and sporicides are used at this facility. Another one could be only 70% IPA is used in the pass-through decon. Concern. Another why could be rinsing with potable water is used each time for the floors and walls. Potable water is potential contamination source, right? That's like tap water. Training on cleaning does not occur on an annual basis. It's every 5 years. Is that frequent enough? Even if you have outside contractors cleaning your facility, would you say training every 5 years is adequate? Probably not. Are they tested? I know Anne-Marie Dixon always recommends not only training annually, but to do some type of testing with them. The mop heads look soiled and black in color, right? And after your use in the clean room, I can understand them being dirty, but how are you laundering them? Are you autoclaving them? Are you steam sterilizing them or gamma radiating them? Why do I ask those questions? Those mop heads get dirty over time, whether they're microfiber mop heads or polyester or foam mop heads. And those mop heads then can be a potential source of bioburden. So that could be a risk. So here's your big concern in cell and gene therapy, ATMP sites, and that is the inside of these BSC hoods. Organisms like molds can grow on the walls in the hood. They can grow on the floor surface of the hood. The other area that you need to make sure to use that sporicide on besides the walls of it and the bottom of it is the outside, the glass and that steel. You need to use the sporicide very frequently, and you need to have that rinse step with the IPA to remove any residue. Why? It's a big risk of spreading fungal spores and bacillus in the BSC hood. So keep that in mind. You want to limit the risk. So what are some of the big concerns from the regulators related to mold? One of the biggest ones I put out here, it came out really recently in a FDA 483 in the chat room. This is where they took finger dabs on an auger plate from an operator in an ISO 5 clean room. And what they found was he was contaminated with mold. So they found Chaetomium and they found Aspergillus. And this was on the fingertips of a gloved operator. So what does that mean? That means that we got some red flags going on there because he was in that aseptic area, and that's a very high risk area for production. And whether we're talking about a compounding pharmacy, or in this case, cell and gene therapy, very concerning and high risk there. Let me go back here. One of the big issues that I've seen this in Rio de Janeiro, Sao Paulo, Brazil, and on the West Coast in the US, are clean room facilities where I look up and I see black and brown stains on the ceiling in the clean room. Those black and brown stains are actually mold growing in the clean room. I've seen that several times and I get very concerned about that. Over the course of the pandemic, we've had a couple of big pharma sites in the US where pipes have broken up above the clean room or behind walls and flooded the room with water. Then I see facilities that decide the way they're going to clean it up is they're just going to throw a number of disinfectants at it and assume that the disinfectants will kill the mold. And that doesn't usually work out very well. So I would recommend in those cases looking at VHP, vaporized hydrogen peroxide, fogging, or doing a triple clean with a disinfectant twice followed by a sporicide to get that room back in control. Triple cleans are very commonly used in the US. You can use a quaternary ammonium twice followed by the sporicide, and that's a great opportunity for you to gather some in situ validation data with the disinfectants to take that data initially when the site's really dirty, and after each phase of the triple clean to show the robustness of your cleaning and disinfection program. And we would refer to that as a field trial and in situ validation. And I can tell you FDA regulators like Thomas Arista and many other regulators, even Andy Hopkins with MHRA, really like to see that kind of data because that shows you under a worst case condition, I can control that bioburden in the room. I've got at least a couple of publications out there in the industry on this, and that's a very hot topic here lately. This is a situation where an FDA regulator walked in the facility and he saw some filled products there, and he actually observed them. Some of these vials were actually had moldy cap lines on them. So moldy caps. Would that be a concern? Absolutely. Because in the US in 2011, we had a big issue come up in the US at the New England Compounding Pharmacy that was totally contaminated with molds, including Aspergillus. So as a result of the mold, it was quite unbelievable. Those mold spores actually got in filled product and went out to customers, so patients, and there were quite a number of deaths from it and injuries. So fungal meningitis was spread, and it created quite a problem in the industry. Now, as a result of that, now the FDA is going into all the compounding pharmacies and regulating them more. So 503B facilities are very frequently visited now by FDA regulators. This is another good example here, and this has deals with HEPA filters. So clumps of dark material that the FDA saw and around the HEPA filters, and I've seen it around gaskets and in light fixtures. That dark material is actually mold. One of the clean room sites that I worked with in Lyon, France, I was up above the rafters looking down into the clean room, and I saw them using water for injection to clean the curtains around a filling zone, and the bottom of the curtains was all black, and the water they were using, the WFI, was black. We all know water for injection should not be black. So I went down and took a closer look at that, and actually I saw it on the floor too, mold spores. It was all Aspergillus growing in the facility. Very big concern. So mold can lead to product recalls. We have seen that. And in some cases with IV bags, so IV bags where they're using to produce saline solutions or antibiotics are always a big concern in the industry. And a lot of times what happens is that saline or IV from the IV bag or the antibiotic will leak out of it and get onto the conveyor belt and make it very sticky, which is a very hospitable environment for mold growth. So in this example, Cladosporium, which is a fairly common mold that we see, especially in Puerto Rico, that got created a lot of contamination in that IV bag site. Big concern there. And this is a good one from a compounding pharmacy. So again, I mentioned that earlier, a lot of the compounding pharmacies seem to think 70% IPA kills everything, and we know that it doesn't. So in this example, the FDA is in there and they're looking at, wrote a warning letter on this, and they're saying there's a lot of microbial contamination from bacteria and fungus in the facility. So that's a big concern. We see that very frequently in the compounding sites now. Why are we so concerned about molds? What is it if I just have one hit of mold? Is that something we really need to investigate? Or should we be trending mold over time? Well, I would propose to you, you need to trend the mold over time, because periodically you are going to get hits from it, just like bacillus. It's not like you're only going to see one mold hit every 10 years. You are periodically going to see it. It's been an issue at several of the sites I've been to in Korea and in China. So molds can be, many of them are pathogenic. Many are toxic. They cause really bad acne. There's some mold strains like Stachybotrys that can deteriorate neurons in your brain, right, and create an issue. And it can certainly infect products that you're making and cause for basically adulteration of the product and having to recall products because of mold. We have seen that. In fact, if you are following the COVID vaccine production, there was a big warning letter out on Emergent Biosciences, and they had to halt their production. They are no longer making the COVID vaccine because one of the things they were doing was dragging bags full of waste through the different clean rooms they have, and it was leaking. So then they had kind of an endemic mold and bacillus issue, and they got written up by FDA on that, and they never did get it completely cleared up. So that's a big concern. So I would recommend looking at your site as a whole, so holistically, making a risk map diagram. Where have you seen the organism? Have I seen it in a mulch pile? Have I seen it in a sterility testing suite in the aseptic area? Where have you seen it? And then, as my good friend in the industry with Pfizer, Amy McDaniel, would say, you really need to speciate it. So the genus and species is very critical to know because we need to know, where are we seeing it, right? Is it the same strain in the sterility suite as in aseptic production? I need to know that information. As Thomas Arista would say from the FDA, what are possible assignable causes? And they need to make sense. So the assignable cause really has to be something that really correlates to your source of contamination. The overall objective should be to holistically, you want to prevent that mold from reoccurring. And it could be from multiple sources. So it's not just that you want to prevent it from occurring in one area. Maybe it's multiple areas, right? But you need to stop it. So that's a key thing to prevent it from coming around again. Because I can hit the area all day with a sporicide or VHP, but if you have not eliminated the source, so if you've not eliminated the sources, that mold will keep coming back. And that's something we definitely don't want to happen. So when you're looking at mold, right, and contamination from it, you should be generating a CAPA investigation on it, especially if it's at very high levels in the ISO 5 grade A area. You want a thorough CAPA investigation, and you can use a process called HACCP, where you're looking at the different hazards that could potentially affect it, right? Where am I getting the hits from the mold? And you should be tracing that to the different areas. So here's a diagram, a table that kind of walks you through that process of, what do we want to look at in a mold investigation at our clean room? Well, looking at the water is a key thing. So if you have a WFI still and WFI system, how often am I sampling it for organisms? What does that data look like from my sampling? So how often am I seeing excursions in there? You may want to know that, right? That's a key thing. Keep that in mind. When we talk about pure steam, right? Is that pure steam potentially contaminated, right? If it is, that's a concern. As I mentioned, some of these organisms that can fluctuate between anaerobic and aerobic environments, you might pick it up in pure steam or in compressed gases like Cutibacterium acnes, right? So how often am I sampling that? Compressed gases, helium, nitrogen, argon. What about the environmental monitoring and data collection? So active air sampling, swabs, subtle plates, what does that data look like, right? How often am I doing it? One of the big sites Neil and I have worked with in Korea, South Korea, around Icheon, they get about 100,000 environmental monitoring samples a year. That's a lot of sampling. They have two entire floors of folks that are going in doing that kind of environmental monitoring. What about personnel? How often am I sampling them? What does that data kind of look like? Those are all things you want to think about. When it comes to cleaning and disinfection, this is our famous pyramid in the industry. So the most frequently used product in any clean room, use about 4 to 5 times as much of it as you do sporicides, is the alcohol. 70% IPA, 70% ethanol, and you would be using this to sanitize items you bring into the room. In fact, Donna Reber from Pfizer, one of my colleagues in the industry, she often recommends always using that IPA on items you bring into the grade A area. So from the ISO 7 area outside the BSC hood and cell and gene into that ISO 5 or grade A area. Why should it be IPA? Because up until then, you're probably using a sporicide. You can also use a sporicide on it, which I like to recommend, but you should use something to control bioburden before you put it into that really critical zone. Then we have disinfectants. This is what the FDA and Thomas Arista from FDA calls the workhorse of your program. So this would be phenolic disinfectants, quaternary ammoniums, used on a routine basis to sanitize the clean room and to control bioburden in the clean room. And that's a key thing because it does two key things. It both cleans the surface and provides a moderate level of kill on the surface. And you really need that cleaning action, even when doing a triple clean to remove soiling, microbes, and dead microbes in the room. Up at the top is what we call the workhorse of your program 2 for any potential spores. So when I need to nuke the area to remove and kill any remaining organisms, I would use my sporicide. So hydrogen peroxide, peracetic acid chemistries, anything like that. So bleach at high concentrations, would be used. So Dan, my colleague, Dan Klein, he's going to take over here. Here you go, Dan.
All right.
And you just tell me when to flip the slide.
Sounds good. So for this section, we're going to talk about the kind of the who, the what, the where, and the how of fungi in cell and gene therapy sites. So the who is, what are we dealing with? What fungal species are problematic? What types of fungi? The where is, where does it come from? How did it get there? And then the what is, what makes it so problematic? And then finally the how do we kill it? How do we get rid of it as something that's very critical, of course. So when we're looking at molds that are common to cell and gene therapy clean rooms and cold rooms that we've seen, here's a list of some of the species that we've identified quite a bit of. Aspergillus is one penicillium species. Jim mentioned the Stachybotrys, the sick building molds, as well as the Cladosporium, on down to Chaetomium, a very challenging fungus, and then even some of the single cell fungi, the yeast. And it's important to note, if you look at the table here, fungi and fungal spores are very hard to disinfect. You go from the bottom of this list, which is the least resistant, envelope viruses, all the way to the top, which includes spores and prions. You can see that the fungal spores are harder to kill typically than gram-negative bacteria. So species such as Aspergillus and Penicillium that produce those conidiospores that are evolved to spread long distances can be very hardy and very challenging to kill with disinfectants. Next slide, Jim. And this is some research that we just is currently in press that we've just published, and it's in the process of being published in a German journal from some testing that we developed internally, in which we wanted to really look at the different fungal species, because not all fungi are the same in terms of disinfectant susceptibility. I mentioned the single-celled yeasts are very easy to kill. They're very much like bacteria when it comes to relative resistance. But when you start to get to the multi-cellular fungi, they start to get more challenging as you try to deal with that kinetiospore. That kinetiospore is a tough nut to crack and can be hard to kill. So you go higher on this list up to the penicillium, which can be very hard to disinfect. And then Aspergillus, which is very tricky. And then Chaetomium, which we're finding is probably one of the hardest to kill fungal spores out there. This also relates back to why it's so important to, as we were talking about earlier, why it's so important to identify down to the genus and species level. So not only is it beneficial to do that ID work, to do that identification work, to see where it's coming from and where you're finding it, and if you're finding the same fungi in different areas, but it's also important because it can give you some clue into how to disinfect that and what you're dealing with. Because if you're dealing with something like Chaetomium or Aspergillus, you might have to use some more powerful interventions than if you're dealing with something like a trichophyton, for example. So the next talks about where. So where do these come from? Typically, fungi are not going to be typically human commensal microorganisms. Humans can have yeast within their body, of course, but typically, unlike Staphylococcus or Micrococcus that's going to be on human skin, fungi are going to come from an external source. They're going to come into the clean room from somewhere outside or get into an area that you're not successfully finding and disinfecting. So some sources that we can talk about are some high impingement spraying devices that cause building damage. Things that we mentioned in terms of broken pipes. If you have water, you're going to have the possibility of having some fungi and be hard to track down. HVAC systems that are turned off, for example. It's a great place for fungi to form and to bloom, and then to spread very easily. You can see in the image there of the Aspergillus, you have the fuzzy top of the kinetiospores, and those get up in air currents and they get from place to place very easily. So those are some possible sources within the ISO 5 clean room. Again, got in there from an external source and established residency and established a contamination source that continues to be problematic. Some other areas in ISO 7 that we've seen, some storage rooms, cold rooms. Fungal spores love things like cardboard. They love things like wood. If any of those are present outside and maybe a storage area that's then brought into a more critical space, can certainly be an issue. So looking next, we have one of those case studies. This was an example of a high impingement sprayer that poked over 30 small holes into the walls, just too much pressure, too much force, and that damaged the wall. And it exposed that drywall, and drywall can be somewhat porous, it can get damp, and that can be a great place for fungi to hide out, and very hard to find and then eradicate. The next example is a kick plate, where behind that kick plate, there was some moisture trapped, and there were fungal spores that developed a contamination behind the kick plate. So between the metal kick plate and the door, there were fungal spores. Every time somebody kicked that door open, those fungal spores got up in the air and got out as a contamination source. Another famous example is the tip of a felt marker. Fungal spores can certainly live on areas like that and can be picked up on that marker and then easily transferred to the next area that the marker touches. The next one's a great example that Jim talked a little bit about, and that's cartwheels. Cartwheels are going to be brought into the area, so that's why it's so critical that anything coming into the area, because again, the fungi are not inherently on human skin, like Staphylococcus epidermidis, for example. So they're coming in from somewhere, and you don't want them to take hold and establish a contamination source that's going to be hard to get rid of. So one thing that you can do, one intervention that can be implemented, is something like disinfecting cartwheels. Spray that thing with a sporicide. Make sure you get 360 degrees all around the wheel. Roll it over a sporicidal wipe for an adequate contact time, for example, or even have a small depression or trough in the floor that the wheel can sit in to disinfect before going in. Those are some very, very key controls and would have solved some of these Aspergillus investigations that we've seen in the past. So that talks a little bit about where they come from, in addition to what we're dealing with. Now I want to talk a little bit more about what we're dealing with in terms of what makes the fungi so problematic. And these are some great SEM images that were taken in, on Aspergillus brasiliensis. And you can see here, you have that mature phase of the spore, of the kinetiospore, the spiny spores of Aspergillus. And these can be very hard to penetrate with disinfectant. Just looking at them under a scanning electron micrograph, you can see under SEM that they're very intact, very rigid, and that can be very hard to disinfect. The next slide shows that they can very easily hide within small cracks or small crevices on a surface. Even a crack that you can't necessarily see with your naked eye can harbor a lot of these Aspergillus spores. And the characteristic of these spores, of these spiny spores, they're very easily hid, but they also aerosolize very easy. So they can get up in the air and travel on air currents from place to place. And if that happens, you get something like the next slide, where things can get out of control very quickly. Because fungi are so good at spreading from place to place through air currents by forming those spores, those kinetiospores that can float from place to place, fungal contamination can get out of control very quickly. You can have a problem very quickly. And you want to avoid situations like this, where your last resort is going to be just to tear everything down to the studs and start over, because it can certainly, fungi can form anywhere you're going to have a little bit of moisture or coming in from the external environment. And then once it gets a foothold as a contamination source, it can spread throughout a room very quickly, very easily, and cause a lot of problems. So how do we kill them? This is some testing that we performed on Chaetomium globosum side by side with Aspergillus brasiliensis. And you can see using a phenolic disinfectant, we got some activity against the Aspergillus. This is a suspension time kill method, so a pretty easy study of using a volume of the disinfectant to which we added the fungal spores. And you can see we got some pretty good activity there with the Aspergillus brasiliensis at both 5 minutes and 10 minutes. But you look at the Chaetomium, and you can see that's much harder to kill. Very challenging microorganism. Again, reinforces why it's so important to have those identifications available, because it can be a completely different animal in terms of something that you have to worry about killing. On the next slide, we compared using just an unformulated quat, so a quaternary ammonium compound active ingredient that was not put into a disinfectant, was just diluted in water. And we wanted to test different fungi just to confirm which ones were the hardest or the easiest to kill relative to Trichophyton interdigitale, Candida albicans, and Aspergillus brasiliensis. So Candida albicans on the right side was very easily to kill in 2 minutes and 45-second contact time using this suspension-based study. The single cell fungus was quite susceptible to disinfection, as was Trichophyton interdigitale. Trichophyton interdigitale is the fungus that's used to get fungicidal claims with the US EPA. Fairly easy to kill. But then you look at the Aspergillus brasiliensis in this case, and about less than a half a log reduction in a suspension study after as much as a 10-minute contact time. So that shows how it can be, fungi can be very different depending on which type you're dealing with, and why Aspergillus in particular can be very, very hard to kill. It's also important to understand your facility, in addition to what fungi you're finding, what fungus has been isolated, because different surfaces can have different results. In this case, we looked at glass and the fungicidal activity of 2 phenols, 2 phenolic disinfectants, and 70% IPA. And we tested penicillium and Aspergillus. And you can see pretty good log reduction values. Glass is a very flat, inert surface. So we got good kill on this surface with the different disinfectants. On the next slide, we looked at stainless steel. Saw some similar results. A little more challenging for the first phenol against the Aspergillus, but pretty decent activity. Again, a fairly inert, fairly flat stainless steel surface, not too many of those places for the conidiospores for those fungal spores to hide. So we had pretty good data there as well. And then for the third set, we looked at a floor material, and we saw worse results with the Aspergillus brasiliensis. So we assume that in this case, we had some small holes or pits within the flooring, natural variation in the floor that made it harder to get the disinfectant in contact with the spore, the fungal spore, and reduce the kill of those two phenolic disinfectants. So it's important to know what fungal species you're dealing with and know your facility, as those are some key variables that can really impact your test results, and comes back to that how do we kill them? Very important to understand. Another key variable, moving on to the next slide, and a critical item to ensure that you have efficacy on how to kill these fungal spores, is contact time. Again, those fungal spores in that cell wall, they have chitin, right? They have glycoproteins, there's pigments, there's a very, very tough cell wall associated with fungi. So you have to make sure that you have adequate contact time of your disinfectant, in addition to choosing the right disinfectants, but have that adequate contact time to penetrate all of those components of that fungal cell wall in order to get kill. This is very important scientifically, but it's also very important from a regulatory perspective because it's a key regulatory requirement. On the next slide, you'll see an FDA recent FDA 483. So this is very recent in 2021. And this again talks about contact times of disinfectants used for gloved hands and surfaces are not documented as reviewed as a disinfectant, and surfaces does not have an established time in procedures. So again, that established time being a key in this 483. And the next 483 related to time and making sure you have that adequate time to get effective disinfection. This one talks about, again, ensuring the contact time is met. This is from 2020, another very recent 483 that we found, where it says you have to ensure contact times are accomplished. There is no contact time established and nothing in the procedure determining how much area can be wiped with a single mop surface or hand wipe surface. And we'll talk more about the technique factors as well. And these things can get quite serious. As I said, fungal contamination can get out of control very quickly. So it can be a point of emphasis for some inspectors. The next slide shows a warning letter in which this talks about adequate contact times for sporicidal agents used as part of your disinfection program for the aseptic processing area, and that your firm failed to establish an adequate system for cleaning and disinfecting the room. So very serious scientific and contamination outcomes can occur if you don't know that who, that where, that what, and the how of how to get rid of fungal contamination in cell and gene therapy, but also from a regulatory perspective as well. Finally, this topic is on contact time, and making sure that you're getting the contact time for hard to kill microorganisms like fungi is something that's getting more attention. This is a very recent article published by a couple of our colleagues in October called A Refresher on Disinfectant Wet Contact Time, that really talks about this topic and the fact that if you're going to validate in a wet contact time, then you have to ensure that you have sufficient liquid volume, sufficient contact conditions to make sure that you meet that validated time. So with that, I'll turn it back over to Jim to talk about techniques and cleaning techniques and methods.
All right, my favorite thing, Dan. So now we'll talk a little bit about cleaning methods and ineffective cleaning methods, right? So let's talk about why some of these things are so important. Well, that's something actually FDA regulators do look at. So here, if you look at this recent 483 by the FDA, they say here that your firm failed to conduct adequate cleaning and disinfection of the aseptic area was a BSC hood. No additional cleaning or disinfection and environmental sampling was conducted in the hood. So proper cleaning technique and use of contact time is something that FDA and even Thomas Arista with FDA will highlight very frequently. I know he has a good example he talks about where he's used a stopwatch to monitor the wet contact time that a facility had for disinfection. So regulators do look very closely at that. This is another recent 483. It says cleaning agents used to disinfect, clean, and sanitize equipment and production areas. of non-sterile product were not suitable for use. Your firm was using expired disinfectants, expired sterile disinfectants. Is that good? Not a good idea, because then you have to do an investigation into that. I'd always recommend I would take a close look at that environmental monitoring data in that area where the expired product was used to see if you have any outlier data that was in that area. Here's another good one. And so this is another recent 483 observation by FDA. Hard to reach areas used to close the direct compounding area within ISO 5 are not cleaned, right? For example, I observed a lack of cleaning by the sterile operator in the upper half portions of the area of the sterile drug production. So probably the operator's cleaning the hood, but not cleaning the top of the hood, not cleaning outside the hood. So that's something I can tell you regulators will definitely watch. So this is from an article that Joe McCall and I published in the industry a couple of years ago in the pharma industry. One of the things we wanted to take a look at is how cleaning methods really affect overall efficacy. So here, you actually take a black light and you can highlight organisms and essentially bioburden potentially on the surface in a room. Well, one of the ways you can clean it is like Dan cleans his Maserati car, right? So if you're going to do the wax on, wax off there, you're spreading that bioburden around. You're not actually cleaning the surface. That's a problem. We always recommend to use unidirectional overlapping strokes by roughly 2 inches or 20% to completely cover the surface. And as you can see here, it's doing a really good job at removing any particles or potential bioburden left on the clean room surfaces. That's the objective. So proper mopping with a microfiber mop, proper wiping are really paramount to having a good and effective cleaning and sanitization program. So again, here we talk about those overlapping strokes. Why do I want to have the overlapping stroke? Well, I want to remove any potential bioburden from the surfaces. I want to make sure I'm killing it. That's a key thing. Just think, every time I get on an airplane, I fly on airplanes all the time, the dirtiest area of the airplane is that seat pocket cover right in front of you. It's loaded with tissues and everything you can think of. And if you were to take Rodax samples of it or swab it, it's loaded with bacteria. So you need to be careful on airplanes too, right? It's all about control. So this is the last topic I wanted to address in the cell and gene therapy talk, because it's something that does come up a lot. So what if I have a big contamination event? Or what if I've had construction in my clean room, or a power outage, or a big, well, let's say a big shutdown for 2 weeks? How do I bring the area back online? Well, this is something me and my colleague there in Singapore, Richard, have published on. And I have actually published some additional articles on it, something Dan and I are working on in the industry, some publications on. We would do a triple clean to bring that area back into control. So what is a triple clean? I outline it here. Take the environmental monitoring data at time 0, see how dirty the facility is after that worst case event. Then you can use a low, as an example, a low pH phenolic or a quat, right? Then you take the T1 environmental monitoring data to show how effective that quat use was. And it probably won't kill everything, because you're going to have some mold spores and bacillus that will not be killed by it. Then you're going to use, let's say, a high pH phenol in the area with cleaning and disinfection. So microfiber mops, wiping. Take T2 EM data and want to take a close look at that data. And then finally, I want to come in and nuke the place with my disinfectant, my sporicide, after that last disinfection step. So the sporicide, let's say it's a hydrogen peroxide peracetic acid chemistry or a bleach at a high concentration. We want to remove and kill everything in the area with that sporicidal step. So finally, at T3, my last EM monitoring data in the area, hopefully all my numbers are 0 or at least greatly reduced from what they were back at T0. That's the objective. FDA regulators love to see the data. So Thomas Arista, John Metcalf, any regulator in the industry, Andy Hopkins, they love to see this kind of data. So what does it look like schematically? I'll show you here. First of all, I want to mention there's no unified definition of a triple clean in the industry, but what I like to refer to it as using a disinfectant 2 times, followed by the sporicidal application. And in some parts of the industry, Anne-Marie Dixon recommends a 9x clean, which also will produce the same kind of results, where you use a sporicide once, followed by a disinfectant twice. And you would do this 3 days in a row. She calls that a 9x clean. I've seen it done in Korea and in parts of New Jersey. It works. You could do a fogging with the sporicide, pre-clean the area with disinfectant, with a surfactant-based disinfectant, and then fog the area. You could come in and use VHP, so pre-clean with disinfectant, and use vaporized hydrogen peroxide. Works great too. But the most economical procedure is to use that triple clean procedure. So a disinfectant 2 times, some sites may do it 3 times, and then bring in that great sporicide to get the reduction of any remaining spores like Bacillus cereus, which could be serious if you get it, or Penne bacillus, or other types of mold, or bacillus. So here's how it looks at time 0. Look how contaminated my clean rooms are. My goodness, wow. Lots of bacteria, fungus, and spores in the rooms. So I want to collect my EM data and show how busy the area is with bioburden at the time. So I want to show, what does the room look like under a worst case condition? So we just had construction, or Dan was doing some cleaning and a pipe burst in the room. Now we're really contaminated. Then we start with our cleaning. So you go from the most critical zone where maybe we have a BSC hood and cell and gene, and you work your way out of the room. You never go from the gowning area into the aseptic area. You go from that aseptic area and work my way out. Then the next key thing is after you start that cleaning, you would want to use the disinfectant. So let's say a quat or a phenol chemistry twice. That, as you can see, really helps to reduce that bioburden in the room because all of my bacteria are gone and almost all the fungi are gone. Really nice data. I'm just left with some really pesky spores left in the room. So then I come in and I nuke the place with my VHP, with my hydrogen peroxide peracetic acid chemistry, or my bleach at a high concentration, and voila, as the French say, I have all zeros. Now you may not get all zeros, but let's face it, you should see a vast reduction in bioburden in the room. That's the real objective here. And when you see that, you know that you're meeting your criteria of really being able to keep that clean room under control, which is what we all strive for here. So finally, these are all the references that Dan and myself use today. There's a whole litany of them. Any of the ones not copyrighted, we're happy to share with you. If it is copyrighted, we can refer you to where you can purchase it online. And with that being said, Linda, we're happy to take any questions.
Fabulous. Thank you, Jim. Thank you, Dan. We'll spotlight Dan as well. And if you want to stop the slide share.
Sure. Let me just figure out how to do that. Hold on a minute. I'm getting my expert level tonight. Stop. How do I stop? Yeah. Sharing screen. Oh, stop share. There we go.
Brilliant. So now we can see you both fully. So we already have some questions for you from Michelle Peak. Question for Jim. How should you measure contact times? Calibrated stopwatch or some other method?
My experience has been in the industry that most facilities will use a calibrated stopwatch. So click it like Tom Arista does, or if you have operators and you have a clock that's easy to read, that's kept good time on the wall, you can mark it down on your log sheet, the time you started cleaning and the time you finished in the room. And my experience has been in ISO 7, ISO 8 rooms, which are grade C and D. When you mop those with the sporicide or disinfectant, they typically remain wet with microfiber mops about 20 to 30 minutes. And then in your ISO 5, grade A, B areas, It's a much shorter time, so you may need to reapply it. And it's a shorter time because of all the airflow in the room. So you might have anywhere from 100 to 800 air changes per hour in there. So you may need to reapply it or do supplementary coupon studies at a shorter contact time to show that you can kill the hardest to kill bioburden in that area. And either of those are accepted by the FDA and MHRA.
Absolutely. The calibrated stopwatch and calibrated timer, still the gold standards, I think, for making sure you ensure that wet contact time.
So, hi both. Sorry, it is Michelle here. Hi Jim, I'm a bit sorry about that.
No problem.
I'm from Australia. We're obviously regulated by the TGA, but dealing with some facilities in Singapore with the HSA, and a lot of my clients.
Great to have you, by the way.
Oh, thank you. A lot of my clients in Singapore and Australia really question the calibrated stopwatch. Is that necessary? So I just wanted to see if that's an industry standard, and I can go back to them and say, yes, it's an industry standard. But thank you for bringing out the comment on reapplying. So a lot of my clients are using pre-impregnated wipes. So they just open up the packet, chuck it onto the mop head and everything. So I would say, as you said, with a lot of air changes in the room, I don't know how wet those pre-impregnated wipes would actually be. So we'd need to look at studies on that.
Right. And normally your vendor, I know we do have some wipes out there as well. We'll have some studies on the saturated contact time, but I will have to say in some of those BSC hoods and ISO 5 rooms, you're talking about a lot of air changes per hour. So you might want to do some studies to support that.
Okay. Thanks both.
Thank you.
Thanks for your question, Michelle. We have a question also from Eva Leung, which is, would it be recommended to take the T0, T1, T2 data required if the T3 data meets the classification limits?
What I normally suggest on that is if you're doing that triple clean, so the in situ field trial initially, I would collect as many data points as you can. But after you've done that initial round, so let's say you have a shutdown twice a year, I don't think you need to do it every time. I do agree with you that you could just do it initially with the dirty facility and after the sporicide step and just really show the effectiveness there. I think when you're doing it initially, you want to kind of show the regulators, hey, each step here, we're seeing some reduction. And finally, at the end, we are killing either everything or hopefully close to everything. Right, Dan?
That's exactly right. That's exactly right. Useful data to have after that T1, T2. That baseline data, though, every time is critical to see what you started with versus what you ended with. But it is nice to have that story of here's my disinfectant, here's my disinfectant, here's my spore side, and this was the impact over that course of treatment.
Thank you. And please do keep the questions coming, or feel free to unmute if you wish to ask either Dan or Jim directly. In the meantime, Anita Naya asks, what would you recommend for cleaning procedure, double bucket or triple bucket system?
I would say both of those are very commonly used in the industry, probably double bucket a little bit more, but I think, whether you go with Micronova, Perfex, Contact, Valeda, Hydroflex, all of them are well known in the industry, and double bucket is probably the standard. Now, I do go to some countries like Brazil, where they use a lot of one bucket systems, and I'm not a fan of one bucket systems. Number one, because that mopping solution gets very dirty very quickly, and you're spreading it around like the dirty water at the McDonald's. And the other key thing is with that single bucket system, you're generating a lot of waste from hundreds of mop heads each time.
I hope that answers the question. Smitha asks, is it mandatory to conduct the disinfectant efficacy study?
Dan, you get this one.
Yeah, you will want to conduct a disinfectant efficacy study. Obviously, you want to leverage any data that's available, but you want to characterize those disinfectants in your facility with your in-house isolates, any problematic surfaces. So you want to collect your environmental monitor isolates, build your library, and test and show that those disinfectants work under the conditions in which they're being used in your exact facility. So yes, you do want to do DET testing. Jim and I are actually putting together another webinar on DET testing specifically. So you will want to have your reference strains, your reference surfaces tested, and then you want to do your in-house isolates and any in-house surfaces. You have to show beyond what the supplier is going to test for registration, whether it's with the US EPA or in Europe or with the TGA in Australia, for example. You want to show, in addition to that, how it works and qualify that in your facility by doing that DET testing. Then, of course, the triple clean in situ stuff comes after that, and then finally the environmental monitoring. And you could argue that the triple clean is really your best example that you have things under control with your disinfection program. But as that baseline, you do want to do that DET. Jim, did you want to add?
Yeah, the only thing I wanted to, great answer, Dan, fantastic. Linda, I just wanted to say that not only are we doing that webinar coming up for STERIS, where we're expecting a lot of attendance on that, we'd be happy to come back and do that disinfectant testing webinar for your ISPE chapter.
Sure.
Thank you. Yes, that would be great. And also on that, as Neo had posted, please do let us know what other topics you'd all like to hear about so that as we plan for next year, we can include that in. So we have got the link for the survey on the chat right now. So just click on that and you can fill that in later. In the meantime, we've got a few more questions, so please do keep putting them in. We've got 3 minutes to go. So Albert asks, with the current supply chain crunches happening everywhere, there would be instances where organizations may be forced to replace disinfectant wipes for cleaning program. What kind of data should be collected to implement the change if the replacement is not a like for like, i.e. different chemistry, for example, amine replaced by ammonium quat? I hope I got that right.
So I think so the question was about supply chain, right? And we were talking about what if you can't get the wipes, is that right?
It's more if you can't get, say you're using an amine wipe and you had to replace it with a quaternary ammonium wipe.
Okay.
So you're going to a different active. So that is tricky. And of course, yes, you have to use what you can get, right? But that is going to be tricky if you have to switch, if I'm understanding the question correctly, if you have to switch to a whole new chemistry, that you will want to do some kind of in situ study to validate that new product is going to be effective. Jim, do you have any approaches to how you tackle that one?
Yeah, I would suggest that, because if you're going to a totally different chemistry because of supply chain issues, like let's say from a alcohol to a quat or an alcohol to a sporicide wipe, you would want to at least collect, if at the very least, some environmental monitoring data with that new method, with that new chemistry, to show that it's effective and that you have some data to support it. I wouldn't necessarily suggest doing a DE study to support it, so a wipe study, but I would suggest collecting some EM data, through maybe swabbing or Rodak plating or even active air sampling too in that area.
Yeah, include that as part of an assessment of risk, because you are changing chemistries entirely. Understood that maybe not an option to do anything else in that case, but that's something you have to tread carefully.
Okay, and on that, Edwin has asked the question, from the regulators, are we expected to set EM alert and action limits specific for mold numbers, and how can we set the limits?
So my experience has been, when you collect that environmental monitoring data and trend the data over time, I would include mold in that. So in other words, I wouldn't just trend the bacteria, I would also look at molds that you're seeing. Because what I hate to see is work with a site where they just have one mold hit, let's say every month, or one hit every 3 or 4 months, and then they do a big Kappa investigation on that, and then they have all these Kappas going on, and they can never get them closed out. So I would look at molds, and let's say, for example, in an ISO 7, ISO 8 clean room, maybe you set a limit of 10 or 20, but because you can have organisms there, it's not where you're supposed to have 0, but I would look at establishing a limit there with the molds, as well as your vegetative bacteria. Now in ISO 5, that's a different story. There, as Dan knows, you're expected to have either 0 or 1. and you could debate that all night long. Right, Dan?
And we have.
Okay, and from I know we're just at the 11 o'clock hour now, but if you'll just allow us to ask the last two questions, if that's okay.
Yeah, sure. And I apologize for that, Linda. I like to talk and sometimes just ramble on.
Oh, no, that's fine. We, in fact, will give the contacts so that if people have any other questions, they can ask you as well, or they can send the questions in to us. But in the meantime, Smitta asks again, how about the disinfectant efficacy study for a new facility? E.g. in Singapore, no isolates and no specific guidelines from local regulatory side, i.e. HSA. Can we follow USP standards?
Dan, you get this one.
Again, it's a question for the local regulatory authorities, but I certainly think that the USP 1072 pass-fail criteria and DET criteria are good references to have. I think it's also important to note with a new facility, you're obviously going to take some time to gather your environmental monitoring isolates. So it could take 6 to 12 months before you even have any bugs that are available to test. But yeah, in the, I would default to whatever local guidance you have. But in the absence of that, I think, yeah, that's a good place to go.
Yeah, and I'll add a couple of pieces of feedback into that. The reason you really want to wait 6 to 12 months with a whole new facility, instead of just using historical data or testing just American type culture strains, is because then you may, if you do it right away with, let's say, reference strains, and then do it again in 6 months with your environmental isolates, you could be paying a fortune for that. It could be a very expensive venture. So that's one thing. And then another thing is you should check with your manufacturer of your disinfectants. Many of them, like us, will have reference strain data that you can use to support the use of the products at your site. You can also do and generate some in situ field trial data until you have enough isolates to be able to actually do that validation. And I think Dan agrees with that.
Absolutely.
And one final question, what would be the frequency to do the DE study - requalification annually with in-house isolates?
I think you want to trend the data at least annually, if not every 6 months, to analyze it and then determine off a risk-based strategy whether or not you need to do additional DET testing. If everything's under control and nothing's really changed and there's no new bugs popping up, no new microorganisms being discovered, no new surfaces being implemented or added, then you might not have to redo the testing every year. But you need to look at your EM data and assess that at least annually, if not twice a year or more. Jim, did you want to comment?
Amen to that, Dan. And I would say that about every 5 to 7 years, you should take a closer look at all of your, essentially all your coupon test data and your field trial data, because one of the key things you want to look at is if some of this old data, and there was actually a recent 483 that came out from the FDA, I'd be happy to share with anyone that's interested, let me know, where they came to a clean room facility and said, look, your data is 10 years old. Have you decided to take a look at that? Maybe some of your controls, aren't right in this. Your log reduction numbers may not have been met. So they had some really old, antiquated data. And the FDA said, no. You need to repeat this study and do it with some current methods in the industry.
Yep.
Okay, brilliant. Well, thank you both, gentlemen, for spending your evening with us. And to everyone who's taken part today, we want to thank you for joining us. For those who joined later, and also those who didn't, who were on time, we also will be sending you the links to this recording, as well as for the PDF slides. So if anyone has any queries as well, you can also come back to us. But in the meantime, we want to thank everybody for taking part. We hope that you've all found this informative as well as enjoyable. And it's always a pleasure to have the STERIS team sharing with us. So thank you both. Have a lovely evening. And to everybody, be safe, stay well, And we look forward to seeing you all again at our next session. Please do give us your feedback. There's a link on the chat. If you could also let us know your details if we contacted you, because we've got a few joiners who we don't know where they were probably referred by their friends. Please do let us know your email so that we can then send you the link to the details. Thank you all, everybody, and take care and have a good day.
Thank you very much.
Thank you,
Bye.
Linda. You're the best.
Thank
Absolutely.
you.
Guys, bye.
Bye.
Bye-bye.